Welcome. In this video, we will address a critical aspect of in vitro cell culture, cell death. Unanticipated loss of cell viability can compromise experimental outcomes and indicate underlying issues in technique, reagents, or environmental control. We will review the major forms of cell death, outline common causative factors, and provide practical strategies for effective diagnosis and prevention. Cell death in mammalian culture systems occurs through several mechanistically distinct pathways. The most frequently observed are apoptosis, necrosis, and autophagy-related cell death. Apoptosis is a tightly regulated, ATP-dependent process marked by chromatin condensation and formation of apoptotic bodies. Necrosis involves uncontrolled cellular swelling and membrane rupture, often resulting from acute damage. Autophagy, while typically a survival mechanism, can lead to cell death when excessively activated. In addition to these, other forms of regulated cell death have been characterized, including ferroptosis, apoptosis, necrosis, and anochies, each involving distinct molecular pathways and triggers. While less commonly encountered in standard culture conditions, they may become relevant depending on the experimental context or cell type. Unexpected cell death in vitro is typically multifactorial. Among the common causes is microbial contamination. Bacterial and fungal contaminations are usually detectable by changes in culture turbidity, pH, or microscopic observation, whereas mycoplasma contamination is more insidious, often going unnoticed under routine observation, but capable of significantly impairing cell viability and function. Media quality is another critical factor. This includes degradation of labeled components such as L-glutamine, improper pH buffering, expired serum, and inconsistent lot performance. Environmental instability, including fluctuations in temperature, CO2 concentration, humidity, or oxygen levels, can lead to stress-induced apoptosis or necrosis. Mechanical stress, such as vigorous pipetting, excessive centrifugation, or incomplete detachment during passaging, may also damage membranes and induce lytic death. Finally, cell density is often overlooked. Both overconfluency and underseeding can disrupt homeostasis and trigger cell cycle arrest or death, particularly in anchorage-dependent lines. Diagnosing the cause of cell death begins with a systematic approach. Start by examining the morphology of the cells under the microscope. Key indicators like membrane blebbing, floating cells, and chromatin condensation can signal apoptotic processes, while swelling or rupture suggests necrosis. Next, perform a viability assay. The trypan blue exclusion test is a reliable method for quantifying live versus dead cells. Live cells will exclude the dye, while dead cells will take it up. Check the pH and color of the culture medium regularly. A yellow discoloration typically indicates acidification due to cellular metabolic activity, whereas a purple hue suggests alkalinization, often resulting from carbon dioxide loss or improper buffering. Ensure that the pH remains within the optimal range for your specific cell type to maintain viability and function. Environmental checks are essential. Verify that the incubator's temperature, carbon dioxide levels, and humidity are stable and within recommended ranges for your cells. Finally, consider routine testing for contamination, particularly for mycoplasma, which often goes undetected under standard microscopic observation. PCR-based assays or mycoplasma-specific detection kits are recommended to ensure cultures remain free of such hidden contaminants. Once the immediate cause of cell death has been identified, corrective actions should be implemented promptly. Begin by replacing any compromised reagents. Always use freshly prepared and properly stored media and serum, and filter sterilize when necessary. For improved stability and reproducibility, use stabilized L-glutamine substitutes, such as Glutamax, which remain effective under standard culture conditions. If environmental instability is suspected, recalibrate the incubator and ensure adequate gas exchange and humidity. Monitor temperature and carbon dioxide daily, particularly after maintenance or relocation. Handle cells with care to avoid mechanical trauma. Use gentle pipetting techniques, appropriate trypsinization times, and avoid over-centrifugation. To prevent recurrence, maintain detailed experimental records, including reagent lot numbers, cell passage history, and seeding densities. Consistent documentation facilitates pattern recognition and long-term culture health. Finally, practice strict aseptic technique at all times to minimize the risk of contamination. To address cell death effectively, assess the morphological features. Identify contributing factors such as contamination or environmental stress, and apply corrective measures with consistency and care. Thank you for watching. If you found this video helpful, please like, subscribe, and share it with your colleagues. For more insights on cell culture, stay tuned to our channel.